Good morning, everybody. This morning we're going to be talking about section 12.2, and section 12.2 talks about parts of the blood. In the diagram you're looking at right now, we're basically looking at what blood looks like when it's separated out into its three major components. If you took blood from a person, put it in a test tube, and spun it in a centrifuge, it would separate out by weight or by density, and what you would see is that the heavier materials would be at the bottom of the test tube and the lighter materials would be at the top. In this particular case, the formed elements, which are the blood cells themselves, would be at the bottom here in this red area. The middle area is going to be where the white blood cells and the platelets will probably be, and that's the small area, the buffy coat. And then the top layer would be the plasma, which would make up about 55% of the blood. It would be mainly water and dissolved materials that are in it, blood proteins and blood gases and so forth. So those are the main components of blood. Basically, like I said, the smallest component of all the blood would be the buffy coat, which would be that clear coat that would be between the bottom and the top, and that would be consisting of the white blood cells and the platelets. There are three basic kinds of blood cells, and of those three, two of them are located in that area, and they are in the lowest numbers, obviously. When we look at the bottom of that test tube, the dark red area, that's what we call the formed elements. And the formed elements of the blood make up about 45% of the blood, which would be mainly your red blood cells. Okay. The bottom area is going to be very thick, very viscous, and the reason why it's red, obviously, is it's mainly made of red blood cells. Red blood cells are called erythrocytes. As you can see, red blood cells are going to be sort of round with a biconcave center. This kind of darkened area that you see in here is where they're kind of pushed in. It's kind of like taking a basketball that's deflated and pushing in with your hands on both sides of it and creating a disc that is concaved on both sides. So the reason why these cells look as they look is for their function, is so that they can get through small capillary walls to their sites so that they can deliver oxygen to cells in the body. So their structures related to what they do, their function. The red blood cell is anucleate, which means it has no nucleus. And most of you now, having studied cells, are probably thinking to yourself, well, all cells have nuclei. Well, these cells have a nucleus when they're born, so to speak. They have all the components that a normal cell has when they are first developed. As they develop, though, they try to become as efficient as they can in carrying oxygen, and they have one main component that makes up most of the cell, and that's a protein called hemoglobin. So as they mature, they pretty much cast off a lot of the normal cell organelles, and they carry on their energy making in a different way that other cells do. So they don't need their mitochondria. They cast those off. They cast out the endoplasmic reticulum, and all kinds of cell organelles are cast off, and basically they're just an oxygen carrying machine. Their function is to transport mainly oxygen. It says here transport respiratory gases, but the amount of carbon dioxide that's carried by the red blood cell is very, very small. They do carry a little bit of carbon dioxide, but they carry them in a different place in the cell other than on the hemoglobin, as where the white blood cells, or the red blood cells carry their oxygen. By far, there are more red blood cells in the blood than any other kind of cell. In men, there can be anywhere from 5 million to 6 million, depending upon the size of the man, of these cells per cubic millimeter. The cubic millimeter is a very small drop of blood. In women, this number is a little bit lower, about approximately 4.5 million per cubic millimeter. The lifespan of a red blood cell is basically 3-4 months, and then they're destroyed in the spleen, and they're broken down, and then the body uses their parts for other things and gets rid of all the other parts that are not necessary. So they basically are produced as a normal cell. Then as they mature, they lose most of their cell organelles, which makes them basically oxygen carriers with hardly anything else in them other than hemoglobin, which carries the oxygen. And then that greatly reduces their ability to stay alive for more than 100 to 120 days. You can see how complicated the hemoglobin molecule is. There are approximately 250,000 of these complicated molecules in a red blood cell, and each one of these has 4 bonding sites for an oxygen molecule, O2. So if you do your math there, you can see that every red blood cell is capable of carrying 1 million oxygen molecules. So that's quite a bit. Kind of an outstanding figure when you look at what the red blood cell is there for. There's a term that's used for the making of blood cells, and that's called hematopoiesis. I threw it in there because this word usually always comes up when we talk about cell components. And hematopoiesis consists of the making of all 3 of the different kinds of blood cells. Specifically, when the body makes red blood cells, this is controlled by a number of hormones and growth factors, and there's a negative feedback system as to how many the red blood cell production and how many we have, and how many are being destroyed. So we keep the normal number pretty much level. This is just a simple diagram to show you. Here's the bone marrow where red blood cell, white blood cell, and platelet production normally occurs in the shaft of long bones. And they basically start from a cell that is non-differentiated, which means it's just a general cell. And then that cell changes into a specific kind of a stem cell. And then from there, that specific kind of stem cell will turn into a more specific kind of blood cell. Obviously, I'm not going to hold you to knowing all the different kinds of cells. I just wanted you to know that they start out as stem cells, and that's what a stem cell is. It's an undifferentiated, just a general cell, and eventually it changes and changes until it gets down to the specific kind of cell it's going to be. In terms of a red blood cell, it goes through many, many changes. Like I said, it starts out as a normal cell with normal organelles, and through this whole process, it goes down the line and it starts casting off some of its cell components until it gets down to the point where it's a reticulocyte, which means it ejects its nucleus, and then it ejects a few more organelles and becomes just simply a red biconcave disc with hemoglobin in it, for the most part, for carrying oxygen. Our body has a system where it reads the number of red blood cells and makes sure that we don't get too many and make sure we don't get too few. And when we do this, it tells us what precautions to take. Sometimes we have too few red blood cells, which leads to something called hypoxia, which means you don't have enough oxygen. When you have too many red blood cells, this increases the blood's thickness, and that's not a good thing either. There's usually a good balance between the red blood cell production and the destruction that goes on in the spleen. And this is controlled by hormones in the body, chemical messengers, and a number of other things. Why could you be hypoxic? Well, first of all, hemorrhage, which means you have some bleeding somewhere in the body, which would cause you to lose red blood cells, and it would reduce those numbers. And if you reduce the number of red blood cells, you reduce the amount of oxygen that your blood is carrying. You can also have insufficient hemoglobin, which means the hemoglobin doesn't have enough iron in it, which is the portion that actually bonds to the oxygen. If you're low in iron, then you're going to be unable to carry the right amount of oxygen. And in a naturally occurring situation where you're in high altitudes where the amount of oxygen is lower, you have a natural kind of hypoxia. You don't have as much oxygen because there's not as much available. Some of the things that go wrong with red blood cell numbers, I already talked about anemia, is when blood has low oxygen carrying capacity. You can have insufficient numbers of red blood cells, or you can have an insufficient red blood cell in itself that has not enough iron. And, another disorder is called polycythemia, which is where you have an excess of red blood cells, which means your blood is way too thick. And usually this isn't a normal condition. This is usually caused by cancer or sometimes the chemicals that come with cancer. Chemicals introduced into the body which make the red blood cell production too high. I'm sure most of you have heard that athletes do something called blood doping. Specifically, years ago, the world-class cyclist would go and they would remove some of their blood a few days before their event, and they would be tested to see what their red blood cell count was. And then as the body replaced those red blood cells, then before the event, the athlete would put those red blood cells back into the body, which meant they would have a higher oxygen carrying capacity. And this has been banned in athletics. One of the other diseases that we see in terms of red blood cell is sickle cell anemia. This is a genetic disorder that is found in about 1 out of 400 African Americans. And basically what they have is abnormal hemoglobin, which under stress will crystallize, and when it crystallizes, it starts to form abnormally shaped red blood cells that are in a sickle shape. They carry oxygen very poorly, and they also get caught in the branches of blood vessels, and cause a lack of circulation. Very serious disorder. Next thing we want to talk about is white blood cells. Here are a number of white blood cells. If you look at them under the microscope, you can tell that they all look very, very different from each other. Now, obviously, we're not going to ask you to know all the different kinds of white blood cells. I just wanted to give you an idea about how different each of the white blood cells are. White blood cells are known as leukocytes. Okay, and if you look at a picture taken under the microscope, a micrograph, you can see that red blood cells are much smaller than white blood cells, and some white blood cells are bigger than others. So you can see all the different kinds of white blood cells mixed together with this smaller cell here, which is a red blood cell. There are some white blood cells that actually leave the blood vessels, and they are made to do that, and they kind of, through amoeboid motion, they'll creep in and out of tissues of the body through the capillaries, and they do so as a macrophage. What a macrophage is it's a cell eater. It basically creeps around in the tissues, and it tries to find foreign invaders. Excuse me. When it finds foreign invaders, it envelops them and takes them into the cell and destroys them. So diapedesis is literally walking through tissues, and it goes in and out of the capillary walls, seeking foreign invaders, and it destroys them. Destroys any foreign invaders that are in the tissues. White blood cells basically are there for our immune system. They help us to fight off foreign invaders, and they do that in a number of different ways. Some of the white blood cells are going to produce proteins which in themselves will kill off foreign invaders. Others, as I said, are macrophages that crawl around in the tissues and seek out foreign invaders and surround them and break them down. A number of white blood cell diseases are evident in a human being, one of them being leukopenia, which you've already looked at in the lab that you did. This is an abnormally low white blood cell count, and usually this has to do with drugs that are taken for other diseases. Leukemias are a cancer of the white blood cells, and we know we have leukemia when our white blood cell count is abnormally high. Normally, white blood cells are somewhere in the neighborhood of about 8,000 per cubic millimeter, and they can get much, much higher than that, 3 times that. And doctors know then that we have some kind of either an infection going on, or in the case of leukemia, it's a way, way out of control, abnormal number of white blood cells. Another disorder is called mononucleosis, which is commonly called mono. And this is when we have an excessively high number of monocytes and lymphocytes because of the presence of a specific kind of virus that causes a fatigue, sore throat, and a number of other secondary infections. The third kind of blood cell that we find in the blood is what we call a platelet. And the unusual thing about platelets is it starts out as one of the biggest cells in the body. It's called a megakaryocyte. And what happens is somewhat similar to what happens in a red blood cell. It kind of goes through a metamorphosis, a change in form, to where it starts out as this huge cell, and eventually, as it matures, the cell fragments and breaks down into cell parts. Okay, and then these cell fragments are then used to plug up holes in blood vessels when there's bleeding. The number of platelets per cubic millimeter is about 250,000. So if we were to rank numbers of blood cells in the blood, number 1 would be red blood cells, which would have anywhere from 4 to 5 million per cubic millimeter. Then it would be platelets that have 250,000 per cubic millimeter. And then it would be white blood cells who have anywhere from, say, 4 to 8,000 per cubic millimeter. So the platelets' main function is to circulate around the blood, and whenever there's a breakage in the blood vessel, it's going to be used to clot the blood there and stop the bleeding. Once again, there's a normal stem cell development that we go through in the hematopoiesis, or cell making of these platelets. Whenever we have a breakage in a blood vessel, which we call bleeding, basically we have tissue damage, and whenever tissue is damaged, it gives off a specific chemical. And whenever it gives off a specific chemical, it causes platelets to stick to that area of the damage. There are a number of chemicals then that are put out, that are not really put out, but are changed. One of them is a fibrous protein that's in the blood that normally does not come into play until there is damage to a tissue, and this tissue damage chemical is put out. This fibrous material then will cover the damaged area, and then platelets will come in and plug up the areas between this fibrous mesh that's put up. So there's a cascade of things that have to occur in order for us to actually heal a broken blood vessel. So you can see here, we break a blood vessel. Basically, the blood vessel starts to constrict to reduce the amount of blood that's escaping. And then the platelets are going to be attracted to that area because of that chemical that's given off. They'll plug in the area where there's a break in the blood vessel. And then those fibers, those protein fibers from the blood, will start to appear, and they will kind of fill in and make a meshwork along with the platelets and red blood cells. So we form a little plug there that stops the bleeding. Usually in a normal bleeding situation, it's stopped within 3 to 5 minutes. If it doesn't, then there's probably massive damage that surgery is required on. And that's what a blood clot looks like up close. We have platelets, red blood cells, and proteinaceous fibers that form this meshwork. Okay. Sometimes we have problems in some of those steps in the clotting process, and whenever we have a chemical, either genetically or because of some kind of other physical abnormality, these chemicals aren't there. One step in the process is taken out, and the blood does not clot properly. Sometimes the body produces too many platelets due to an inflammation or infection or even a cancer, and this is called thrombocytosis. Whenever we produce too few of them, this is called thrombocytopenia. Too few platelets is caused, obviously can be caused from bleeding. It can also due to suppression or destruction of bone marrow. Sometimes when people have a cancer, they have radiation, and sometimes this can depress the production or the hemostasis or hematopoiesis of the platelet production. One of the main diseases that we hear of, which is also a genetic disorder, when we have an inability to clot our blood, and that's called hemophilia. That is a recessive, sex-linked genetic disorder. And you can have several different forms of hemophilia depending upon which chemical is not being produced because of malformed genes in your chromosomes. Whenever you have hemophilia, you basically bleed for long periods of time. You have trouble clotting your blood. And when this happens, literally a person can bleed to death. Usually people that have hemophilia are going to receive those chemicals on a regular basis that are missing because of that genetic disorder. So we treat the disorder by filling in those missing clotting factors in your blood. Okay, so those are the parts of the main parts of the blood. We have the plasma, and then the formed elements. And the next time we talk, we're going to be talking about different kinds of blood. Blood can be typed some 74 different ways. We're going to talk about one of those ways, actually two of them, the ABO blood system and the RH factor. So the next time we talk, we'll be talking about blood types. And that'll be all for today. Adios, amoebas.